Good morning, everybody. Thank you for being here. Welcome to Grand Rounds. This is the this Grand Rounds is in a special location, obviously, because we are linked today to the first of what we hope will be a really successful series of annual translational science retreats meant to highlight the amazing resources that are present at Yale Cancer Center for people who do translational science, and also to highlight some of the amazing stories that have come out of this work. And so no one better to be our inaugural speaker than Dr. Katie Politi. Katie studied biology at the University of Pavia in Italy, and then moved to New York, obtaining her PhD in genetics at Columbia University. She then joined Harold Varmus's lab at Memorial Sloan Kettering and began her life's work on the molecular basis of lung cancer. She continues this work at Yale now as a professor in the departments of pathology and internal medicine in the section of medical oncology. Her laboratory is focused on studying the biology of lung cancer and uncovering mechanisms of resistance to targeted therapies and immunotherapies in this disease. She's also got a keen knowledge of essentially every mutation that's ever been described in lung cancer. And I know that doctors often call her up and say, what drug should I use? She co-leads the Cancer Signaling Networks research program. She's the scientific director of the Center for Thoracic Cancers, co-director of the Yale SPORE in lung cancer, and recently elected to the AACR board of directors. So we're really appreciative that you're going to kick us off today. The ID number there is to record your attendance, and then we'll have questions both in the room and online when we're done. Thank you.
Thank you very much, Barbara, for that wonderful introduction. And thank you very much for having me as a speaker today. It really always is, I think, very special to speak at one's own institution, and then especially also associated with this first translational science retreat. So I'm really excited about this. And today what I'm going to do is I'm going to tell you about some of the work that we've been doing over the past few years in the laboratory. These are my disclosures. So we have a long-standing interest in the lab on studying lung cancer. And as all of you know, there are several histological subtypes of lung cancer. But one of the things that we've learned over the past 20 or so years is that lung cancer is not one entity, and that there are, in addition to different histological subsets of the disease, there are also a variety of laser pointer of molecular subsets. And in particular in lung adenocarcinoma, through various sequencing efforts, different mutations in genes that encode either receptor tyrosine kinases or downstream signaling components of receptor tyrosine kinase signaling pathways that regulate cell proliferation and cell survival have been identified, as you can see here in this pie chart. And I think one of the things to really highlight is what we've learned over the years is that these mutations are, in addition to being molecular, to establishing molecular subsets of the disease, they really also are clinically relevant, because different targeted agents have been developed that can be used to block the activity of these mutated oncogenic drivers. And in particular, and in the work that I'll tell you about today, for example, mutations were found 20 years ago now in exons encoding the kinase domain of the epidermal growth factor receptor in about 15 to 50% of lung adenocarcinomas, depending on which population you look at. And these are mutations that confer sensitivity to EGFR tyrosine kinase inhibitors. But there are many other targeted therapies as well. So you can have rearrangements in the anaplastic lymphoma kinase and targeted therapies that are effective in that, and so on, for a number of different oncogenic drivers in lung cancer. And so this has really transformed the field. And so if we look at this diagram here of approved FDA approvals for lung cancer, in recent years, what you'll see is there really has been an explosion in FDA approvals, especially from the early 2000s, in the 2010s, and approvals now also in the first part of the 2020s. Most of these agents that were approved recently have been targeted agents, and that really is linked to the discoveries of these molecular subsets of the disease. But also, I think one of the things that has been emerging also in the past 10 to 15 years really are the approvals of immunotherapies that we hear a lot about, agents that are targeting immune checkpoints like the anti-PD-1, anti-PD-L1 axis, and CTLA-4. And so this has really been transformative in lung cancer. And I'd just like to point out how in recent analyses, what we're seeing is that there's actually a decrease in mortality from lung cancer in recent years. And in a study published in the New England Journal of Medicine a few years ago, it was really shown that the decrease in mortality from lung cancer can't be accounted for just because of a decrease in incidence of the disease, but is likely reflects actually advances in the care and in the new therapeutics that have emerged, including in particular in the years that were studied in this paper for targeted agents. And so I think this is a really nice example of how what we've learned over the years from the biology and from the genetic studies of tumors really is having a profound impact for patients with this disease. And of course, I would be remiss if I didn't point out how immunotherapies have also been transformative. And I think the continued decrease in mortality that we are continuing to see is actually going to show how it isn't only the targeted therapies, but also the immunotherapies that are really contributing to this decrease in mortality from lung cancer. So if you look at this, there's really, these advances have been tremendous, but what we do know is that both primary and acquired resistance to targeted therapies and to immunotherapies are common. And here you can see an example of scans from a patient with tumors with a KRAS G12C mutation treated with a KRAS G12C inhibitor, and you could see the tumor regresses, but then comes back, and you have these as acquired resistance. And here, if we look at this plot taken from a review looking at studies of immunotherapies, you can see that across various different indications, but including in lung cancer here, that in clinical studies of immunotherapies, the response rates to immune checkpoint inhibitors are not super high. We're not talking 70, 80% the way we're talking with some targeted therapies. Not only that, but also we see acquired resistance commonly emerging. So there's a lot of work that needs to be done to really understand and optimize treatments for both targeted agents and immunotherapies, and to understand mechanisms of sensitivity and resistance to these agents. And so what do we do in my lab? And as part of the research program, we are really interested in understanding mechanistically biological processes that are involved in cancer. We like to integrate these with studying and addressing clinical challenges and investigating specimens and data from patients with cancer. And really the hope is that the work that we do collectively as a group, this is work that we do with many different people, is to discover findings that will lead to clinical trials and new therapeutic approaches to patients. Central to our research program is the use of biological specimens from patients and analysis of these specimens. And I think this slide is also going to be showed later in the day as an example of one of the resources that we have as part of the lung cancer program, to really be able to collect and use specimens from patients. And this is just one of the examples of one of the resources. I think you'll hear about a couple of others later on as well, but really an effort that started many, many years ago, working initially with Scott Gettinger and Anna Wirtz and Roy Herbst and many, many people in this room, now with Sarah and many of all of the thoracic oncologists on the team and pathologists, Kurt, for example, really working on collecting specimens from patients who have advanced lung cancer through treatment, especially at the time of resistance, so that then we can take these specimens and analyze them, generate patient-derived models. And really, these have contributed extensively to the work that I will tell you about today. And so I put a little cryovial here. And so what I'm going to do through the talk is when you see a cryovial on the slide, it actually is an example of data that we've been able to analyze and use because of the specimens that were collected through this approach. So you'll see that throughout the talk. So what am I going to tell you about today? So I think, as most of you know, we have a longstanding interest in studying the biology of EGF receptor-driven lung cancer. And so when patients, and really the focus that we've had, at least in the past or until recently, has really been, and because of the sort of the clinical landscape, has really been on advanced metastatic EGF receptor-driven lung cancer. And so when patients are diagnosed with EGF receptor-driven lung cancer, now they're mostly treated with tyrosine kinase inhibitors, most recently, and in the United States, especially the tyrosine kinase inhibitor, osimertinib. This is one of the newer generation of agents that has more activity on mutant EGFR compared to wild type, so hopefully decreasing its toxicity, and has been shown to have superior progression-free survival and overall survival compared to standard of care earlier generation tyrosine kinase inhibitors in this disease. And so this was a really important advance in the field. However, what we do know is that still resistance or acquired resistance to osimertinib occurs almost inevitably. And it actually isn't very commonly associated with on-target EGFR mutations. And this is different from some of the earlier generations of tyrosine kinase inhibitors that instead, where we saw commonly one most frequently observed on-target EGF receptor mutation, the T790M mutation. But you see additional mechanisms of resistance, MET amplification, for example, so a bypass signaling pathway being one of the more common. Then we see histologic changes in the tumors that occur quite frequently. But then most of the mechanisms of resistance are really not known and poorly understood. And so one of the things that we've been interested from as we think about these problems is really understanding these tough challenges, like really understanding this part of the pie chart, right? What are these mechanisms of resistance? What is happening in these tumors where we don't really have a key genetic alteration that is changed, that or clear process that is happening that we can target. And so just a couple of thoughts that sort of guide our thinking. Targeted agents are probably not sufficient. We need to discover new untapped vulnerabilities of oncogene-driven lung cancers. And then the tackling resistance requires new knowledge of the links between cancer cell plasticity, the tumor microenvironment, and tumor heterogeneity. And so these are some of the, and so I think of these as like the not the low-hanging fruit, but the fruit really at the top of the tree that we're trying to really grasp and understand. When we, and really, if we look at EGF receptor-driven lung cancer and we think about it, one of the things that we know is that with the targeted agents that I've told you about today is we do see this acquired resistance, but not only that, we also know that when we use the targeted agents, they don't completely eradicate all the tumor cells, and there's variability in the depth and duration of responses in patients. And you can see this really in this waterfall plot where there's some tumors that shrink dramatically and others that shrink less. And so we've been interested in the question of what accounts for this heterogeneity in disease progression and sensitivity to tyrosine kinase inhibitors. And so the first thing that I'm going to go through is some of the work that we've done to study how different EGF receptor mutations can actually have distinct properties. And so first of all, I've sort of told you about EGF receptor mutations, and one could think, oh, we could lump them all together. But in reality, what we do know and what is becoming, I think, increasingly clear in recent years is that there are different EGF receptor mutations. And not only that, the different EGF receptor mutations have different properties, both biological, biochemical, and also in terms of TKI sensitivity. And so when we look at EGF receptor mutations, there are two major categories of mutations. There's the L858R point mutation, and then there's a set of small in-frame deletions, some of them more complex in exon 19. The most common of these is this E746 to A750 mutation, but then there are these other indels that are found at variable frequencies in these tumors, but they exist. And so what does it mean? Are all these mutations alike? Well, one of the things that we know is that even if you just broadly categorize the L858R mutations and the EGFR exon 19 deletion mutations, and you look at the survival curves on osimertinib from the trial of frontline osimertinib, you see that even just the exon 19 deletion mutations, the overall survival is about 40 months in that study, but for the L858Rs is about 33 months. And this is consistent across different tyrosine kinase inhibitors that are used. So the L858R subset does worse with TKIs compared to the exon 19 subset. We also found several years ago in work that we did together with Sarah Goldberg and Mark Lemon, is that there's a small in-frame deletion in a proline insertion mutation, and one of the exon 19 deletions that actually, if you look at that mutation and you look upon treatment with erlotinib, this was a few years ago, so one of the early generation tyrosine kinase inhibitors, that the progression-free survival, duration of treatment, overall survival were all worse for erlotinib in that subset compared to the more common exon 19 deletion mutation. And this, along with some laboratory studies, really piqued our interest in studying these differences a little bit more. And here you see the cryovial up here. This is also work that Zenta Walter was really central to helping us identify these patients for this study. And so working with lots of different groups here, we were able to show that this proline insertion, for example, what you see in Western blots is when you treat with tyrosine kinase inhibitors, it's less sensitive to various tyrosine kinase inhibitors compared to the canonical EGFR exon 19 deletion mutation. Not only that, when you actually go and look biochemically, and this is work that was spearheaded by a former student that Mark Lemon and I shared, Iris van Alderweireld van Rosenberg, showing that this proline insertion mutation has a KM for ATP that is more closer to the wild type, in contrast to some of the other variants that instead are more sensitive to tyrosine kinase inhibitors. So really it's that affinity of the kinase for ATP that is probably rendering it more resistant to these tyrosine kinase inhibitors. So really from the clinical observations, from some of the laboratory studies going to the biochemistry, we were really able to figure out what was happening with this variant. And this led to work that we did together with Mike Grant and Sarah Goldberg really putting together a multi-institutional cohort of patients with EGFR exon 19 deletion mutations treated with osimertinib, because we wanted to look at the tyrosine kinase inhibitor that was really clinically relevant for patients right now and that was being used to see what outcomes were for patients with this proline insertion mutation with osimertinib. It's pretty rare, so you have to really work together and put together a cohort from various institutions. And so Mike and Sarah assembled this cohort, including data from our Yale cohort, and actually showed that in patients whose tumors have this proline insertion mutation treated with osimertinib, you have worse progression-free survival than if you look at the common EGFR exon 19 deletion mutation. The overall survival isn't quite statistically significant, but you can see that there is a trend in worse outcomes there as well. And so what does this mean? What does this make us think? I think the message here is that not all mutations are the same. And now we have the tools and the drugs to better match mutations with therapies. We aren't the only ones who are thinking about this. There's some other work from Jacqueline Robichaud and John Haymack's group at MD Anderson, work from Christine Lovely at Vanderbilt, all really pointing in this direction. We need to know about the biology, the biochemistry of the mutations, and that can help us think about perhaps how to better optimize these therapies now that we have them. Another point, the structural and biochemical understanding of the effects of the mutation can guide predictions for TKI sensitivity and resistance. And of course, the other question that comes along is, how do we translate to the clinic now? What are the next steps that we can take? So we can test trials of like optimal TKIs. So now we have all these reagents, we can test other agents and other drugs on these different variants and see if there are some that are more effective for specific mutational subsets. But then the other question is, are there other agents that we should be thinking about for certain subsets of the disease in combination with osimertinib? And I think this will be a recurring theme throughout the talk. So for example, should we be thinking about specific antibody drug conjugates or other approaches to target tumors that don't do as well with monotherapy osimertinib? So after thinking about the different, so we talked about how different EGF receptor mutations themselves can have an impact and have distinct properties. But what about co-mutations? How can co-mutations influence tumor progression, but also TKI sensitivity? And so many years ago now, I probably started working on this actually almost exactly 20 years ago when EGF receptor mutations were discovered. I think it was May 2004 that I started generating these mouse models. We generated genetically engineered mouse models of EGF receptor-driven lung cancer in which we could express the EGF receptor mutants inducibly in the lung epithelium. And these were really to be able to study the biology of the disease. And we've used these mice extensively over the years to study signaling by mutant EGF receptor, discover resistance mutations to tyrosine kinase inhibitors, identify therapeutic strategies to overcome or prevent drug resistance, and study the effects of targeted therapies on the immune microenvironment. And here you can see MRI images. We use MRI imaging for our mice to look at the lungs and see, you can see lungs full of tumors. You treat them with the tyrosine kinase inhibitors. The tumors shrink and go away. Over time, the tumors come back and we can study those resistant tumors. So a few years ago, we decided to upgrade our mouse model and use a slightly different system that would allow us then also to be able to modulate other genes, because we know that EGF receptor mutations in lung cancer don't occur in a vacuum. There are other mutations in the tumors there, and we wanted to be able to model that. So we decided to take this tetracycline inducible EGFR allele, cross it to another mouse that in which using Cre recombinase, you can then turn on expression of the reverse tetracycline transactivator, which can bind the TET promoter in the presence of doxycycline and induce expression of EGF receptor. And we also crossed it to a p53 floxed allele. But using this mouse, what happens is we can deliver Cre recombinase. We deliver it with a lentivirus into the lungs of mice, turn on mutated EGF receptor. Simultaneously, we can delete p53. And here's some images. These are the lungs of mice. You can see by MRI, you can see here by histology and a bigger magnification of the histology. So we said, okay, so we have this mouse model with now EGFR mutant and p53 deficient tumors. The p53 deficient tumors are higher grade, they're nastier. I see Rob Homer here. He has helped us extensively over the years characterize and study these tumors. And so one of the questions that we had is, well, in addition to p53, what role do other mutations in EGF receptor play in EGF receptor-driven lung cancer? How do they affect tumor progression? How do they affect TKI resistance? And how do they affect the molecular properties and phenotypes of the tumors? And so what we did is we worked with a colleague at Stanford University, Monty Winslow, who had developed an approach and used it in KRAS-driven tumors to really be able to inactivate, using CRISPR-Cas9 technology, different tumor suppressor genes simultaneously in the lungs of mice. So not all of them in the same cell, but you can deliver this kind of pool of lentiviruses. And in different cells, you can then inactivate different tumor suppressor genes. And then you can use a computational approach that he developed called tumor barcode sequencing, which based on various controls that are spiked in and based on barcode IDs, you can actually look and quantify the effect of inactivating that tumor suppressor gene on the number and size of tumors in a screen. It's essentially a way of doing an in vivo screen. And so we applied, we took this pool of lentiviruses targeting different tumor suppressor genes that were frequently altered in lung cancer, not necessarily in EGF receptor-driven lung cancer, but in lung cancer. And he had used it in the KRAS model previously. And so we applied it to our EGFR L850 at our p53 model. And in particular, we had also crossed the model that I just told you about with one that has an inducible Cas9 allele. So these are our experimental animals here. These are controls because they don't have Cas9. You can't do CRISPR-Cas9 mediated genome editing when you don't have Cas9. So we transduced the lungs of the mice, waited 11 weeks, and then took the lungs of the mice and did tumor barcode sequencing. In our control animals, when you look at the relative tumor size compared to controls, you don't really see any, the tumor suppressor gene inactivation doesn't have any effect, but that's because you don't have Cas9, so you shouldn't see anything. So that was reassuring. What do we see in the mice with Cas9? So one of the things that we saw is that when you inactivate APC from the Wnt signaling pathway, RBM10 and RB1, these three tumor suppressor genes, when inactivated, had the biggest effect on tumor growth. So the tumors grew faster when you were inactivating these tumor suppressor genes compared to controls. We also noticed, interestingly, that SETD2 and LKB1, both of these putative tumor suppressor genes, I'd say, actually had a negative effect on tumor growth, which was quite interesting and is, and I'll tell you a little bit more about that in a minute, but it's a topic of interesting work that we're doing. And then there were a number of tumor suppressor genes that really had no effect on tumor growth. We went ahead and we validated these using single sgRNAs. This is towards APC, and this is to RBM10, which is an RNA binding protein and a splicing factor. You can see that when you inactivate them, you see these bigger tumors, and tumors progress faster than in the EGF receptor p53 model. So what does this mean, though, in the context of human cancer? And so if we, what we did at that time is we actually interrogated the AACR Project Genie database, which is a large data set that has a lot of mutational information that has been contributed to this data set from various institutions that are from their tumor sequencing efforts at their institutions. And when we look in this data set at EGFR P53 driven tumors, and we look at the frequency with which there are alterations in this co-occurring tumor suppressor genes, you actually see that the top hits, RBM10, RB1, and APC, were the top hits in our functional screen in mice. So we think that our screen in mice is actually telling us something about the functional relevance of these alterations in the human tumors. And ARID1A didn't come out in our screen at 11 weeks, but we actually did another time point at 19 weeks and it popped up. So perhaps it's more important later in tumorigenesis. And interestingly, genes STK11 is LKB1. It's really not frequently altered. And that was the one that I showed you seemed to have a negative effect in our in vivo screen. So we've actually, this has been a really powerful system, and we've actually been able to do broader screens with more genes and try to learn a little bit more about what genes are important for the progression of these tumors. And I'd just like to highlight an example of work that we did continuing this effort with D2G Oncology, a company that was co-founded by our collaborators, Monty Winslow and Dmitry Petrov, and we worked together on doing this screen of additional tumor suppressor genes in the context of EGFR tumors, but also in the context of KRAS-driven tumors, for example. And, you know, I'd just like to go back to LKB1, for example, showing how this has a negative effect on EGFR-driven tumors. It's not really contributing, it doesn't really co-occur mutationally with EGFR-driven tumors. So it seems to be like a synthetic lethality with these tumors, but it's an amazing contrast with what we see in KRAS-driven tumors, where it is one of the major drivers of tumor growth. And so this is, I think, telling us, and it's frequently mutated with KRAS in human tumors. So we're really think that this is a cool system to be able to understand how co-occurring alterations impact the fitness of tumors. And Fran Exposito in the lab is really working a lot to understand this synthetic lethality and is doing experiments to knock LKB1 out in established EGF receptor tumors and see what happens, and also to understand mechanistically what is happening in these tumors. So stay tuned for data on these studies that I think will be really fascinating. And there are some other targets that we're studying along these lines as well. So I think a very powerful system. We've also used this approach not just to study mechanisms of tumor progression, but also use this type of approach to really understand what genes can modulate the sensitivity to tyrosine kinase inhibitors. So we did the same experiment, and instead of just waiting and collecting the tumors, what we did is we also had an arm where we treated for 2 weeks with a tyrosine kinase inhibitor, osimertinib. You see here the tumors go away, or they're shrinking mostly. They're not completely going away at 2 weeks, but you do see a response. And so we did the same tumor barcode sequencing. And what we found here is, so this is the plot that I showed you earlier, looking at what is affecting tumor growth. But when we add osimertinib, one of the things that we saw is that KEAP1, the tumor suppressor gene KEAP1, that really didn't have much of an effect on the growth of the tumors in the absence of drug, now limits the sensitivity to osimertinib. In other words, the tumors aren't shrinking as much as wild type or control tumors do when KEAP1 is present. What do we think is happening here? Well, we know that KEAP1 is important to sequester NRF2 in the cytoplasm. When you knock out KEAP1, NRF2 can then go into the nucleus and activate antioxidant response elements in those gene expression programs that allow cells to really withstand oxidative stress. And when we take our mice and we just use an individual sgRNA targeting KEAP1, these are the control mice that don't have Cas9. You use osimertinib, the tumors go away. You don't really see anything left in the lungs. But if you have the experimental mice that have Cas9 and you use the sgRNA targeting KEAP1, treat with osimertinib, you see tumors are still left over. And so again, what does that mean for patients? So at the time, what we did is we worked with Jessica Hellier and Heather Wakely at Stanford University, who had a collection of data from patients with EGFR P53-driven lung cancer, and looked at whether there were mutations in genes in the KEAP1 axis in these tumors. And you can see here in this blue line, the patients who had mutations in the KEAP1 axis in their tumors had a shorter time to treatment failure compared to controls, suggesting that if you have alterations in this program, this antioxidant response program, you're going to have limited sensitivity to tyrosine kinase inhibitors. And so I think one of the things that we're really seeing emerging from this work, looking at the tumor suppressor genes, is that when you do have mutations or you have alterations that co-occur with EGF receptor and with EGF receptor P53, these can modulate both the growth and sensitivity to these agents. We were interested in looking further, and in work that Paul Stockhammer, who was a resident, is now a hospitalist here and is an incoming hem-onc fellow, did recently. He looked at both our Yale internal data from our tissue collection program, you see the cryovial here, but also at the AACR Project Genie data set, and looked at outcomes for patients on tyrosine kinase inhibitors whose tumors had different combinations of mutations. And I think the takeaway here is he was able to look at tumors that had mutations in a subset of tumor suppressor genes, because tumors had been analyzed across a wide variety of different platforms. So we had to sort of focus in on the common subset of tumor suppressor genes that were looked at across platforms. But essentially, if tumors had both P53 mutations and a mutation in at least one of these tumor suppressor genes that he looked at, they had worse outcomes. These are EGFR mutant tumors, even compared to mutations that just had P53 mutations and were wild type for those different tumor suppressor genes. And so what does that mean? Again, I think we're identifying a subset of tumors where there may be a benefit from adding a different therapy, or it should be at least be investigated from the get-go, because they are likely to have worse outcomes with monotherapy tyrosine kinase inhibitor treatment. And this is very relevant right now, at least in the field of EGF receptor-driven lung cancer, because there are studies of chemotherapy plus osimertinib in the first line that are positive, but people are very reluctant to give that combination to everybody. If we can identify people who might benefit more or might need it more, then that could be really helpful for deploying these different strategies in the clinic. And then I think another point is that we're really learning that co-mutations can affect therapeutic sensitivity. And it isn't only in the context of EGFR tyrosine kinase inhibitors. This is happening in multiple contexts and with multiple agents. So here an example, I'm just giving you a few examples. There are many more in the literature, but if we look at KEAP1, going back to KEAP1 alterations seem to have been negative for response rates to sotorasib in KRAS G12C driven lung cancer. Worse, higher local recurrence with chemoradiation. In the context of immunotherapy, LKB1 mutations actually seem to be worse, be worse for, or define a subset that does worse with immunotherapy. And so in conclusion for this part of the talk, the nature of the oncogenic mutation and co-occurring mutations affect sensitivity to TKIs and mechanisms of resistance. We've developed a new generation of genetically engineered mouse models that can be used to study these complex genotypes. And I'd like to point out that really we have a lot of work that is happening now studying these individual different components. Mariana Do Carmo is an MD PhD student in the lab. She's studying the role of RBM10 in EGF receptor-driven lung cancer, working with Luisa Escobar Hoyos's lab, because we really can join forces, and Luisa is an expert in splicing, and this is a really important gene protein that is involved in splicing. So we're doing that. I told you about Fran's work. We have Kita, who's working on KMT2D, which I didn't tell you about, another potential target that came out of this screen. So really, we can really study these different genotypes and understand the biology of these different complex genotypes, which is really exciting. We have found out that inactivation of these different tumor suppressor genes can have different effects on both tumor growth, including positive and negative effects, and TKI sensitivity depending on the oncogenic context. We showed that KEAP1 loss limits sensitivity to osimertinib in mice and in patients, and think that this is really potentially a bad actor if there's KEAP1 alterations, either at the genetic level or also alterations in the pathway. The pathway can be modulated in many different ways. Tumor suppressor gene mutations can be used to identify patients, subsets of patients who are likely to have worse outcomes and could be considered for additional therapeutic interventions. So in the last part of the talk, I'd like to tell you about some other work that we've been doing more recently to study non-mutational mechanisms of resistance, and I'd say also of persistence, so on tyrosine kinase inhibitors. And so what are some of the things that we're thinking about broadly in the lab when we think about this problem of this 50% of tumors that for which we don't know why a resistance emerges? So some of the things that we're really interested in understanding and studying are how the tumor microenvironment affects resistance and persistence. And this is work that we're doing collaboratively, Jake Schillo in the lab doing collaboratively, working with Don Nguyen's lab. We are studying lineage plasticity and tumor heterogeneity, and I'll tell you about an example of this that was just recently published this month, and that comes out of work studying mechanisms of tumor persistence. And of course, another area that we're really interested in is, well, we've talked a lot about genes and mutations and genetics here, but are there ways of reading out pathways and learning about how pathways are altered in tumors, which might be an important way of understanding resistance and persistence as well. And so one of the non-mutational mechanisms that we recently discovered and published on, I'm not going to tell you about that today because I don't really have time, is that we identified a role for the ATPase of the SWI/SNF complex in mediating resistance to tyrosine kinase inhibitors. And SMARCA4 is actually usually lost. You have loss of function mutations in tumors. One of the things that we found, which was really interesting, is that actually it seems to be important for the resistance phenotype, because in resistant tumors, it actually can promote accessibility of chromatin at both cell proliferation genes, but also at genes that are NRF2 loci, so that allow activation of genes that are antioxidant genes. So it links to that KEAP1 finding that we had in our tumor suppressor gene screen. So I'm not going to tell you about this, but I did want to highlight it as one of the some of the work that we have done recently on non-mutational mechanisms of resistance. What I really wanted to focus the last few minutes of the talk on is telling you about some work that we've been doing to study tolerance and persistence to tyrosine kinase inhibitors. And you saw this waterfall plot earlier, but one of the questions that we've had, and I think that is prominent question in the field is why aren't all cells eradicated upon TKI treatment, right? Because if we could get rid of all of the cells from the get-go, we wouldn't have the problem of acquired resistance. And here are some scans. You see the tumor, and you see several months later, the tumor is still there. There still is some residual tumor left over. So what is the biology of residual disease? Well, we decided, and this is work from a former graduate student in the lab, Bomiao Hu, who is now in Arnaud Auger's lab as a postdoc, and I think Bomiao is here. I thought I saw him. And so what Bomiao did is he took advantage, again, of our collection of specimens from patients, and he said, well, what happens if I implant these PDXs that we've generated, treat them with a tyrosine kinase inhibitor, and then look at residual disease? We can harvest that. We take it at a plateau, right? Once the tumors aren't shrinking anymore, that's what's left over. And it's really hard to study residual disease in patients. We can't really easily do biopsies on treatment, but this is as a surrogate of that. And so here are some examples of the PDXs that Bomiao studied. So he took these PDXs, treated them, and then took what was left over after 4 to 6 weeks of treatment when they plateaued. And you can see in all of the cases, there was tumor left over after treatment, varying amounts of tumor, and in some very little, very small islands of tumor, but there was tumor left over. And I'd like to highlight an example of one of the things that we found from one of these PDXs that we studied in a little more detail. We found that in one of them, we saw upregulation of ASCL1. ASCL1 is a basic helix loop helix transcription factor. It has a role in neuronal differentiation, and its expression actually identifies a subset of small cell lung cancer. So it was really up in the residual disease in this tumor. And not only was it up at the transcriptional level and the signature was enriched in the residual disease, but its downstream targets, RET, BCL2, and DLL3 were also all turned on in the residual disease in that tumor. Osimertinib was working really well. You can see phospho-EGFR is gone here. And so this was really interesting to us because we know that a subset of EGFR-driven tumors, when they're treated with osimertinib, can actually undergo neuroendocrine differentiation and transform to small cell lung cancer, a subset of which are ASCL1 positive. And so this kind of piqued our interest. And so one of the first questions that we had was, are these ASCL1 cells present in the tumor pretreatment? And so when we looked and we did single cell RNA sequencing, we actually saw that if you look at the pre-treatment specimen here in blue, there is a subset of these cells that is present that is ASCL1 positive. So we think that those cells were present beforehand. Whether other cells then turned it on, we can't really tell from the types of experiments that we did, but we do know that there was a population that was there pre-treatment. And so our next question after that was, well, how is ASCL1 conferring TKI tolerance? What is happening? And so we said, okay, let's turn to our human EGF receptor-driven cell lines and let's express ASCL1 in these cells. And so one of the first things that we did, we expressed ASCL1 in the cells. And you can see here in this HCC827 cell line, we expressed it and we saw more colonies. And you can see this quantified here after treatment with osimertinib compared to the empty vector control. We did this across in another cell line and we saw no effect of ASCL1 expression. And so this was also interesting. And we said, okay, so why is ASCL1 has a phenotype in one cell line, but not the other? We did gene expression profiling. And what we saw is that in the permissive cells, these HCC827 cells, you actually saw that ASCL1 could lead to an EMT gene expression program, whilst it had no effect at all in the PC9 cell line. And we went on and we looked with ATAC-seq at chromatin accessibility at EMT genes, and we see that upon ASCL1 expression, you do see changes in chromatin accessibility at both epithelial genes and mesenchymal genes when you put ASCL1 into these HCC827 cells that are permissive, but you don't see any changes in the PC9 cells. And so what do we think is happening? So we think that when you have, when you don't have ASCL1, the TKI can work and you see death of the EGF receptor-driven cells. If you have a permissive cellular context, what happens is that you can have ASCL1 can turn on or can lead to an EMT program, and we know that that is associated with resistance to tyrosine kinase inhibitors. In a non-permissive cellular context, though, you don't turn on this program, so you don't have a difference in ASCL1 expressing versus non-expressing cells. We also found that pre-existing cells with transcriptional features of drug-tolerant cells are present in the untreated tumors. And I think one of the questions that we're really interested in is, why are some cells permissive or not? I think this is sort of one of the major problems in cancer, one of the things that has been a mystery in cancer over all of the years. Why do you see certain phenotypes when you have certain settings and not others? And in the case of ASCL1, this is very reminiscent of reprogramming, because it's known, for example, that you can put ASCL1 into fibroblasts and reprogram them to neurons, but when you put them in keratinocytes, you can't. And this has been shown to be due to actually the chromatin landscape at ASCL1 target genes in the different cells. So could something like that be happening in the cancer cells as well? And one of the other questions, of course, that we have is, since ASCL1 is important for neuronal differentiation, it's associated with neuroendocrine differentiation, is it poisoning these cells? We didn't see any other, neuroendocrine markers on, but is it poisoning the cells to undergo that type of change? And so, some of the things that we're thinking about now, and we have experiments ongoing, we have Mark Wiesehofer in the lab, who's been thinking about this and working on this in the context of both prostate cancer, where very similar things happen, and lung cancer. We're asking, how does the chromatin state of a cancer cell affect responsiveness to therapy and plasticity? And so you can have these different cells, you add ASCL1, and you can see different things happen in these different cells. And why is that happening? And is there something that we can learn from these cells that then we can apply to human tumors? And could we use this information? I'm thinking a little bit far ahead, but it's something that's in the back of my mind, is can we predict how a tumor will evolve on treatment with this knowledge? So finally, a couple of final thoughts. So what have I told you today? Baseline mutations and co-mutations can affect disease progression, drug sensitivity, and mechanisms of drug resistance. And how can we incorporate this knowledge into clinical investigation and practice? This is something that we think about a lot. There's a vast heterogeneity and complexity of non-mutational resistance and persistence mechanisms, and we're working to identify them, establish when they are relevant for specific tumors, and find vulnerabilities of these, and be happy to talk more about these thoughts throughout the day today. There are a lot of people to acknowledge. Here are some pictures of lab members throughout the years. Here's a particularly fun one. This was a fundraising picture for a Closer to Free team. So I thought that was pretty cool. These are our Halloween, one of our Halloween parties, and other pictures from all of the lab has contributed tremendously to all of these efforts over the years. And I'm so grateful to have the opportunity to work with so many talented people. There are lots of people to acknowledge who have contributed to this work, in addition to members of the lab. So many collaborators outside of Yale, but in particular, everybody here at Yale, which I hope everybody is on this slide. It's one of the things that I was worried about, but want to make sure that everybody is acknowledged here because of the tremendous contributions that makes it such an amazing place to work together. A couple of things that I'd like to say. We have a retreat too in thoracic cancers on Monday. It's retreat season. It is at West Campus. So you're all invited to join us. We have a team that has been working. Sarah's in here, I think, Sarah Goldberg, Justin Blasberg. We have Glynis Arnold and Melody Noga Mena, who's been working to organize this retreat. So we hope you can join us. And then save the date for our annual lung cancer workshop on June 12th and 13th. It is also going to be at West Campus here. And it's particularly special this year because we are going to be recognizing the 20th anniversary of the discovery of EGF receptor mutations in lung cancer, which has really transformed the field. It's near and dear to my heart, as you can imagine from the talk, but it's really going to be, I think, a spectacular event with lots of people coming from all over to mark this moment. And so we hope that you can participate in that too. Thank you very much, and I'll be happy to take questions.
Thank you so much, Katie. That was wonderful. Are there questions in the room? Maybe I'll start as a person who knows more about squamous cell cancers than adenocarcinomas. When you talk about P53 mutations, are they always the same in adenocarcinoma patients? And we spend a lot of time in the squamous world talking about disruptive mutations, gain of function mutations.
Yeah, we have, I think there's a wide variety of P53 mutations that you see in lung cancer. So they're like different types.
And have you dissected out if they have different implications? We think the gain of function mutations don't lead to as much genomic instability, for example.
Yeah, those are things that we haven't studied that much. I think Paul had looked at the different mutations a little bit in terms of outcomes, Paul Stockhammer, and I don't think he had found differences in terms of outcomes with TKIs with the different classes of mutations. Fine.
So it looked like in your suppressor gene screen that your biggest hit, at least in one of the assays, was loss of RB. But it looks like in the cancers, RB loss was relatively infrequent. Does that suggest, or have you looked at whether there's other dysregulators of the RB pathway that are more common in lung cancer, like the cyclin CDK pathway, and if that's a potentially targetable approach?
Yeah, that's a great question. So it's interesting because RB, as you said, RB1 loss is one of the biggest drivers of tumor growth in our screen. It is also, if you look at how frequently it co-occurs with EGFR and P53 mutations, it's one of the tumor suppressor genes that is most frequently co-altered. So none of them go really above the like 10% threshold. We do know, we haven't really looked at other ways in which the RB pathway could be altered in tumors. We haven't really looked at that. What we do know is that if you have tumors with EGFR, P53, and RB alterations, those are the ones that have the highest likelihood of undergoing that neuroendocrine differentiation. And so like a quarter of those will undergo the neuroendocrine differentiation.
Any other questions from? Okay, thank you again so very much.
Thank you.